Now for a discussion on glycolysis. But before we actually get to glycolysis, let's do a brief overview of cell respiration as a whole. Cell respiration is catabolic and exergonic. We say cell respiration is catabolic because it's breaking apart complex macromolecules. And when we are breaking apart those bonds that hold the macromolecule together, we release energy. And exergonic means that reaction releases energy. When we discuss cell respiration in cells, usually we use glucose as the macromolecule that we're breaking down. So we're starting with glucose. In the presence of molecular oxygen, we release carbon dioxide and water. Now if we look carefully at the atoms in this reaction, we'll see that the carbon is actually losing electrons. It's losing the electrons that were carried along with this hydrogen atom. So since carbon is losing electrons, it's oxidized, and oxygen is gaining electrons as it's gaining those hydrogen atoms. So it's gaining electrons, so it is reduced. So in cell respiration, glucose will be oxidized and oxygen will be reduced. Since glucose is oxidized, everything else around it becomes reduced. So we call glucose the reducing agent. And oxygen is reduced, so everything else around it becomes oxidized, so we say it's the oxidizing agent. Now let's look at a general overview of cell respiration in a picture. So this picture looks very, very complicated, but we'll be breaking it down bit by bit in the next few videos. So we start all metabolism with glycolysis. So glycolysis is breaking down sugars. So glycolysis happens in the cytoplasm, so out in the cell. And then this orange region is the mitochondrion. So organisms that use an electron transport chain, so those molecules that pass electrons from one to another to increasing and more increasingly electronegative atoms are called our electron transport chain. So if we have a cell that's using an electron transport chain, it will pass the products of glycolysis to the mitochondrion, okay? And it will undergo respiration in the mitochondrion. So respiration involves both the citric acid cycle and then it's followed by oxidative phosphorylation. So the products of glycolysis, if there is an electron transport chain present, those products pass to the mitochondrion, enter the citric acid cycle, and then go through oxidative phosphorylation. If an organism does not have an electron transport chain, and you'll notice we're abbreviating electron transport chain as ETC from now on. So if they don't have an electron transport chain, or if for some reason the electron transport chain is unavailable, for example, if a cell does not have oxygen, which is at the end of the electron transport chain, if it doesn't have oxygen, it can't utilize the electron transport chain. And instead, it will pass the products of glycolysis through the process of fermentation. And fermentation also happens in the cytoplasm. So again, with glycolysis, we make something. If we have an electron transport chain, those products can enter the mitochondrion to harvest even more energy. If we do not have an electron transport chain, those products will instead enter fermentation and recycle the ingredients for glycolysis. Now let's look at glycolysis. Glycolysis literally means splitting of sugar. Lys means split, and this glyco means sugar. So we're splitting sugar. So it cleaves or breaks apart glucose, and glucose is a 6-carbon sugar. Remember C6H12O6, so it has 6 carbons, and we're cleaving it into 2 molecules of pyruvate. And pyruvate is a 3-carbon sugar. So that makes sense. We're going from a 6-carbon sugar to 2 3-carbon sugars. So glycolysis requires an input of 2 ATP to get started. So it requires an energy investment of 2 ATP in order to get started. As a result, it produces 4 ATP. So if you get 4 ATP, you gain 4 ATP, but you're subtracting 2 ATP at the beginning, you have a net gain of 2 ATP. And during the process of glycolysis, 2 NAD are reduced to 2 NADHs. So we're adding a hydrogen to 2 NADs. Okay? So in glycolysis, we're splitting glucose into 2 molecules of pyruvate. So you see we start with glucose and end with 2 pyruvates. So for each glucose, we get 2 pyruvates. Throughout glucose, when we first add our ATP, we use that energy from ATP to phosphorylate and rearrange the molecules of glucose. And we rearrange them into these complex molecules. You don't have to know their names, but I've added them here. Just know we're investing energy to rearrange the glucose into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So these molecules can switch back and forth from one another, but we ended up with since they switch back and forth, we ended up having just 2 glyceraldehyde 3-phosphates. And those glyceraldehyde 3-phosphates enter the second part of glycolysis. So in the second part of glycolysis, we are breaking down the glyceraldehyde 3-phosphates, and we're releasing 2 NADs, okay, and 4 ATPs and 2 hydrogens. You don't have to understand exactly how all of that happens. Just know that we start with glucose, okay, we rearrange the atoms and add 2 ATP to do that. We phosphorylate some of the carbons in here, and then we break those apart, releasing 2 pyruvates, so 2 3-carbon sugars, some water, and then 2 NADs, 4 ATP, and 2 hydrogens. Again, you'll see that we gained 4 ATP, but we had to invest 2 ATP, so we ended up with a net gain of 2 ATP in the process of glycolysis. Now, if we want to see how we actually make ATP in glycolysis, we use something called substrate-level phosphorylation. And substrate phosphorylation is when ATP is produced by an enzyme. This purple blob is our enzyme, and the enzyme mediates the transfer of phosphate from another organic substrate to ADP. So this is our enzyme. It's going to transfer this phosphate from this substrate to ADP. We'll see later when we have oxidative phosphorylation, instead this phosphate is going to be free. It won't be attached to a substrate. But since it's attached to a substrate, we call it substrate-level phosphorylation. So this enzyme removes this phosphate and transfers it to ADP. Remember, ADP has 2 phosphates, but if we add another one, it now has 3 phosphates, and that's ATP. So we've created a new product, which is just our substrate minus a phosphate, and ATP, which provides energy for the cell. So this is how ATP is produced in glycolysis and in the citric acid cycle. And just a little fun fact, glycolysis is hypothesized to be the most ancient metabolic pathway present in modern organisms. It happens in all organisms. So let's do a quick recap of glycolysis. Our inputs for glycolysis is a glucose molecule. Remember, glucose has 6 carbons. We have to invest 2 NADs, those are our electron carriers, and 2 ATP to get started. All right, and our outputs are 2 pyruvates. Remember, pyruvates have 3 carbons each. So for each glucose molecule, we get 2 pyruvates. Okay, we also produce 2 NADHs. They now have gained an electron, so they've been they've gained an electron here, and then we have 4 ATPs. So our net gain of ATP is 4 minus 2, so our net gain is 2 ATP. So the question is, where to next? If there is no electron chain available, we'll go through fermentation next, and fermentation happens in the cytoplasm. If there is an electron transport chain available, we will now enter the mitochondria for respiration.